the emergence of life is something almost impossible. So in the end, how was the very first form of life born? If we throw hundreds of components that could theoretically be assembled into a watch into water, relying solely on the forces of nature, could those pieces ever put themselves together into a functioning mechanical watch? The answer is clearly no. Even with unlimited time, such a situation could never occur in the real world. And yet, the birth of life is a miracle, and the appearance of humanity is a miracle among miracles. The universe itself may be like a gigantic, pre-wound mechanical clock with everything operating according to a predetermined plan. Toward the end of his life, Einstein once remarked that everything in the universe is designed, including the emergence of human beings. This profound contemplation immediately plunges us into the heart of abiogenesis, one of the most stubbornly resistant questions in modern science, a chasm separating the inert world of chemistry from the vibrant dynamism of biology. To appreciate the near impossibility of this transition, one must first grasp the staggering statistical improbability inherent in assembling the simplest functional biological entity from the primordial soup of its chemical precursors. Consider the hypothetical mechanical watch analogy, a compelling reductio ad absurdum against purely random self-assembly. A watch, complex as it is governed by deterministic laws of mechanics. Life, however, requires not just complex assembly but the establishment of self-replication, metabolism, and compartmentalization, the irreducible triad of living systems, all operating far from thermodynamic equilibrium, a state that the second law of thermodynamics, often misinterpreted as a universal downhill slide into disorder, seems to actively disfavor. The Earth, approximately 4.54 billion years ago, presented an environment radically different from our own temperate, oxygen-rich present. The Hadean Eon was characterized by intense volcanism, frequent meteorite impacts, the late heavy bombardment being a significant event that severely disrupted early planetary surfaces, and an atmosphere vastly different from today's, likely rich in methane, ammonia, hydrogen, and water vapor with very little free oxygen, a condition essential for preventing the rapid oxidation and degradation of complex organic molecules. This reducing atmosphere, coupled with abundant energy sources, ultraviolet radiation from the young, energetic sun, lightning discharges, and geothermal heat from hydrothermal vents, provided the necessary kinetic impetus for chemical reactions, acting as the cosmic blowtorch to spark the initial synthetic processes. The foundational challenge, universally acknowledged, is the origin of the information-carrying molecule capable of faithful, yet error-prone, replication. The modern biological paradigm rests on deoxyribonucleic acid, or DNA, a double helix of astonishing complexity. DNA requires sophisticated protein machinery, including polymerase enzymes, helicases, and ligases, to replicate and transcribe its information into functional proteins, which in turn catalyze metabolic processes. This presents the classic chicken and egg paradox: which came first, the information carrier or the machinery to read and replicate that information? Modern biology is trapped in this dependency loop. The prevailing scientific hypothesis that attempts to sidestep this dilemma is the RNA World hypothesis. Ribonucleic acid, or RNA, possesses a remarkable dual capability. First, certain RNA molecules, termed ribozymes, can exhibit catalytic activity, functioning as primitive enzymes to speed up specific biochemical reactions, thereby resolving the machinery requirement. Second, RNA is structurally similar enough to DNA to serve as a template for information storage and transmission. The proposal suggests that life initially operated solely based on RNA, both information carrier and catalyst, before evolving the more stable DNA for long-term storage and the more versatile proteins for efficient catalysis. The plausibility of the RNA world hinges on the prebiotic synthesis of its constituent parts, ribose sugar, the nitrogenous bases, adenine, guanine, cytosine, uracil, and phosphate groups. The Miller-Urey experiment, conducted in 1953 by Stanley Miller and Harold Urey, famously demonstrated that amino acids, the building blocks of proteins, could form spontaneously under simulated early Earth conditions involving methane, ammonia, water, and electrical sparks. While subsequent refinement of the atmospheric model suggests that hydrogen and methane might have been less abundant, modern variations of the experiment, utilizing more realistic volcanic gas mixes, still yield a wide array of organic molecules, including several nucleobases. For example, research published in the early 2000s has shown that ADE9 can form readily from hydrogen cyanide, a molecule readily available in planetary atmospheres and meteorites. However, the synthesis of ribose, the sugar backbone of RNA, remains a significant hurdle. Ribose is notoriously unstable under prebiotic conditions, often forming complex mixtures or degrading quickly. A breakthrough came with the Formose reaction, which can generate a variety of sugars from formaldehyde, but this reaction lacks specificity, producing ribose alongside arabinose, xylose, and others in uncontrolled ratios, making selective polymerization difficult. A more refined hypothesis centers on mineral surfaces, particularly borate minerals. Borates have been shown experimentally to stabilize ribose and increase the yield of ribose relative to other sugars, suggesting that specific geological niches, perhaps near evaporating hot springs or in transient pools, might have acted as natural selection filters for the chemical precursors of RNA. Assuming the monomers, the bases, the sugar, and the phosphate were available, the next monumental step is polymerization, linking these units together to form long strands of RNA. In aqueous solution, polymerization, linking monomers by forming a bond that releases a water molecule, is thermodynamically unfavorable. Hydrolysis, breaking the bond by adding water, is favored. This necessitates a mechanism to drive the reaction forward, effectively removing water or concentrating the reactants. Deep-sea alkaline hydrothermal vents have emerged as a leading contender for the cradle of life due to their strong chemical gradients and surfaces rich in iron-sulfur minerals which can catalyze reactions. These vents provide a continuous supply of chemical energy, specifically proton gradients across mineral membranes, mimicking the electrochemical gradients that power modern adenosine triphosphate synthesis in cells. Further research supports the idea that clay minerals, such as montmorillonite, could have acted as scaffolds. These minerals possess layered structures with high surface areas and internal charges, attracting and concentrating organic molecules. Experiments have demonstrated that RNA oligomers, short chains, can form on the surfaces of montmorillonite, possibly through cycles of wetting and drying, which effectively drive the condensation reaction needed for polymerization by temporarily removing water. This geological process introduces a form of kinetic control over an otherwise unfavorable reaction. Once a short RNA strand formed, the next critical hurdle is the development of a template-directed replication mechanism capable of fidelity sufficient to maintain information across generations, yet error-prone enough to allow for evolutionary variation. This is the essence of natural selection operating at the molecular level. Experiments simulating primitive RNA polymerase activity, often using mineral catalysts or simple peptides, have shown that template-directed ligation, the joining of nucleotides along a template strand, is indeed possible, though efficiency and fidelity are extremely low compared to modern biological polymerases, which achieve error rates in the range of 1 in 100 million base pairs. The early RNA world likely suffered from catastrophic sequence degradation, meaning life could only persist if replication outpaced degradation, even at low fidelity. The transition from a collection of self-replicating molecules to a true protocell involves the crucial step of encapsulation. Life requires a boundary, a membrane, to maintain internal chemical concentrations distinct from the external environment, allowing for the establishment of independent metabolic pathways. Phospholipids, the primary component of modern cell membranes, spontaneously form vesicles in water due to their amphipathic nature, having both hydrophobic, water-repelling, tails and hydrophilic, water-attracting, heads. However, the building blocks for these complex phospholipids might not have been readily available in sufficient quantities on the early Earth. Alternative models propose simpler, potentially more robust membranes. Fatty acids, which are shorter and simpler than phospholipids, also self-assemble into vesicles, or liposomes, under appropriate pH and ionic strength. Laboratory demonstrations confirm that these fatty acid vesicles can encapsulate short RNA strands and even undergo division or fission when subjected to physical stress, a rudimentary form of reproduction. Furthermore, these primitive membranes are permeable enough to allow the necessary small molecules for metabolism to enter, yet selective enough to concentrate the internal components, thus coupling the chemical reactions inside the vesicle with the physical stability of the boundary. This merging of chemical reaction networks within a self-organizing compartment marks the transition from mere chemistry to protobiology. The metabolism within these protocells is the next layer of complexity. It is improbable that the full, intricate machinery of the Krebs cycle or oxidative phosphorylation arose fully formed. Instead, a simpler, autocatalytic chemical cycle must have existed, a system where the products of the reaction catalyze the formation of the reactants, creating a positive feedback loop that drives the system far from equilibrium. Iron-sulfur clusters, abundant near hydrothermal vents, are strong candidates for these earliest primitive catalysts, capable of mediating basic redox reactions, the core of energy acquisition. The hypothesis suggests that energy was harvested directly from geochemical gradients, perhaps reducing simple carbon compounds or utilizing chemical energy released from mineral transformations, long before photosynthesis evolved. The sheer improbability of this sequence, monomer synthesis, polymerization, template-directed information storage, encapsulation, and autocatalytic metabolism, is what fuels the conviction expressed by many, including Einstein, that the universe appears almost designed. If one were to truly model the probability of these independent steps occurring sequentially and successfully in one location on the early Earth, the resulting number would be astronomically small, approaching what is effectively zero within the observable timeline of the planet's habitable phase. The statistical argument suggests that for life to emerge, either the constraints of abiogenesis are far less restrictive than current models suggest, or the process was heavily guided. This leads to discussions that blur the lines between traditional biochemistry and philosophy, touching upon concepts of fine-tuning. The physical constants governing the universe, the strength of the strong nuclear force, the ratio of the electron mass to the proton mass, the cosmological constant, appear exquisitely balanced to permit the existence of complex structures, including carbon-based chemistry and stable stars that produce heavy elements. If these constants were even slightly different, carbon chemistry would be unstable, or stars would burn too quickly, precluding the necessary billion-year timescales for evolution. The anthropic principle, in its weak form, simply states that we observe these values because only in a universe with these values could observers exist to make the observation. However, when applied to abiogenesis, it suggests that the parameters of early Earth chemistry must have been precisely calibrated to funnel chemical evolution toward a biological outcome. The panspermia hypothesis offers an alternative route, suggesting that the seeds of life, or complex organic precursors, did not originate solely on Earth but were transported from elsewhere in the cosmos, perhaps via meteorites or comets. Evidence supporting the extraterrestrial origin of organic molecules is robust. Analysis of carbonaceous chondrites, such as the Murchison meteorite, revealed the presence of over 90 different amino acids, sugars, nucleobases, and fatty acids, all formed abiotically in space. This suggests that the initial chemical hurdle, the creation of the building blocks, might have been solved across the galaxy. If life itself arose elsewhere and seeded Earth, it simply shifts the problem of abiogenesis to another planetary body, but it reduces the statistical improbability on Earth. By utilizing an existing reservoir of complex prebiotic chemistry accumulated over billions of years of cosmic evolution. However, panspermia does not solve the central question of the first spark, it merely relocates it. Furthermore, the journey through space subjects any potential protocells to lethal doses of radiation, particularly UV and cosmic rays, which efficiently destroy complex organic structures. While some dormant microbial spores, like those of Bacillus subtilis, have shown remarkable resilience to short exposure in low Earth orbit, the survival of a truly primitive, non-enzymatic protocell through interplanetary transit remains highly dubious without significant shielding, such as deep within a massive meteorite core. Let us return to the terrestrial scenario, assuming life did arise here, and explore the transition from the simple, non-enzymatic RNA world to the sophisticated DNA, protein world, the transition that cemented life's dominance. The shift from RNA to DNA provided superior stability. Deoxyribose, the sugar in DNA, lacks the 2' hydroxyl group present in ribose. This hydroxyl group in RNA makes the backbone susceptible to self-cleavage, hydrolysis, under basic conditions or even neutral conditions over long timescales, whereas the lack of this group in DNA renders it chemically much more inert, crucial feature for storing genetic information across vast evolutionary distances. The simultaneous evolution of dedicated DNA polymerases and sophisticated protein synthesis machinery, ribosomes, was necessary to cement the modern central dogma. This transition implies a co-evolutionary dance. Perhaps the earliest protocells utilized short RNA strands that could catalyze the formation of short, functional peptides from the available amino acids. These primitive peptides, even if inefficient, might have offered a slight catalytic advantage over the ribozymes, leading to their selection. Once a peptide was formed that could assist in RNA replication or stabilization, a rudimentary polymerase, a chemical dependency was established. Over time, the system would favor molecules that could stabilize these interactions, leading to the development of the genetic code itself, a lookup table translating nucleotide sequences into specific amino acid sequences. The establishment of the genetic code, the mapping of triplets of nucleotides, codons, to specific amino acids, is another instance of extreme contingency. Why are certain codons assigned to certain amino acids? While some correlations exist, for instance, codons rich in purines often code for hydrophobic amino acids, the overall mapping appears arbitrary, suggesting a historical, rather than purely chemical, origin. This arbitrariness reinforces the miracle aspect, a slight shift in the initial chemical preference, or a different set of available prebiotic amino acids, could have resulted in a life form with a completely different, perhaps non-viable, biochemistry. Consider the staggering number of possible chemical reactions that could have occurred on early Earth. For life to emerge, the chemical pathways had to converge upon a specific, functional, self-sustaining loop, avoiding the infinite number of non-productive chemical dead ends. This convergence implies either an enormous amount of time, which was available, but under conditions that also favored degradation, or a strong directional force, that design perceived by Einstein. This perceived design resonates with theories concerning the mathematical underpinning of complexity. The emergence of self-organization, leading to life, is often described through the lens of complexity theory and nonlinear dynamics. Systems poised at the edge of chaos, between perfect crystalline order and complete randomness, exhibit the greatest potential for emergent complexity. Life resides precisely at this threshold. It is ordered enough to maintain information, low entropy within the system boundary, but chaotic enough, high error rate in replication, metabolic plasticity, to explore new solutions through evolution. The transition from chemistry to biology is perhaps best understood not as a single grand event, but as a series of cascading, irreversible evolutionary steps where each stage established a necessary prerequisite for the next, creating what evolutionary biologists call ratchets, mechanisms that make stepping backward chemically or evolutionarily difficult or impossible. The encapsulation by fatty acids was a ratchet. Once established, the system could no longer survive if the membrane dissolved. The adoption of the genetic code was a ratchet. Once a functioning, replicable system was based on that code, switching to a different code would require rewriting the entire functional library of inherited information. The fossil record offers tantalizing but frustratingly sparse glimpses of these earliest transitions. The oldest widely accepted undisputed microfossils date back to approximately 3.4 to 3.5 billion years ago, found in the Warrawoona group in Western Australia, appearing as stromatolites, layered structures created by microbial mats. However, evidence for life pushes this timeline back further. Isotopic signatures in ancient zircons from Western Australia, dating to 3.7 billion years ago, suggest fractionation of carbon isotopes consistent with biological metabolism, favoring the lighter isotope, carbon-12, over carbon-13, hallmark of photosynthesis or chemoautartrophy. More controversially, graphite inclusions in rocks from Iswa, Greenland, dated near 4.1 billion years, show similar carbon isotope anomalies, implying that life might have emerged almost as soon as the Earth cooled sufficiently to support liquid water, perhaps within the first few 100 million years. This rapid emergence, shortly after the late heavy bombardment subsided, strongly supports the idea that given the right conditions, the precise chemical mixture, energy flow, and suitable mineral catalysts, abiogenesis might be a chemically favored, rather than utterly improbable, outcome. If abiogenesis is rapid and chemically likely under Hadean conditions, the miracle lies not just in the first spark, but in the sheer universality of the underlying chemical blueprint across all known life. Every life form on Earth, from the simplest archaea to the most complex mammal, shares the same chirality, handedness, for its amino acids, L form, and sugars, D form, the same genetic code, and the same basic cellular machinery. This homochirality is deeply perplexing. In a purely abiotic synthesis, molecules would be formed in a racemic mixture, 50% left-handed and 50% right-hand forms. For life to function, particularly for proteins that require specific three-dimensional folding, one-handedness must be overwhelmingly selected. The process by which this homochirality was established remains an open question. Leading theories involve interaction with circularly polarized light, potentially in interstellar space or on Earth, or preferential crystallization catalyzed by specific mineral surfaces that themselves possess inherent chirality. The mineral quartz, for example, exists in left and right-hand crystal forms. If a chiral mineral surface selectively catalyzed the polymerization of D-ribose or L-amino acids, it could have provided the necessary filter to select for one enantiomer, effectively biasing the entire chemical trajectory of life toward the configuration we observe today. Once a few functional ribozymes or peptides based on L-amino acids existed, their own replication process would preferentially utilize and generate more of the same, solidifying the homochiral environment necessary for complex biological function. The implications of this universal blueprint are profound. It strongly suggests that all extant life descends from a single, last universal common ancestor, LUCA, which must have already possessed the basic machinery, a functional RNA peptide system. The true, unimaginably improbable origin event likely occurred before LUCA, resulting in the successful protocell lineage that ultimately triumphed over countless failed chemical experiments. LUCA was not the first life, it was merely the most successful survivor of the initial abiogenetic crucible. This brings us back to the concept of the universe as a pre-wound clock, deterministic system where the laws of physics and chemistry, when applied to the initial boundary conditions of the early Earth, inexorably lead to complexity. If the laws are universal and immutable, and if the initial conditions, the presence of necessary elements synthesized by prior stellar generations and distributed via supernovae, were met, then the emergence of life might not be a random roll of the dice but a necessary consequence of physical law, albeit one operating over vastly long timescales and requiring specific environmental niches. However, the deterministic view struggles to account for the vast, branching tree of evolution that follows abiogenesis. While the initial spark might be inevitable given the starting state, the trajectory of life thereafter involves countless contingent events, mass extinctions, geographical isolations, random mutations, that shape the final outcome, including the appearance of Homo sapiens. If one were to rewind the tape of life to the point just before the Cambrian explosion, or even to the time of LUCA, and let it play forward again, the resulting biota would almost certainly be radically different. The dominance of bilateral symmetry, the development of eyes, the specific evolution of tool use, these are products of historical contingency acting upon a basic, self-replicating chemical system. This dual interpretation, the necessity of the initial chemical transition versus the contingency of subsequent biological evolution, is where the ore resides. The initial emergence requires the universe to be set up correctly. The fundamental forces must allow for stable carbon chemistry and water chemistry. The subsequent path requires random variation filtered by environmental pressures. Looking forward, research continues to probe the limits of non-biological complexity. Synthetic biology aims to create artificial protocells. Scientists have successfully created minimal genomes, stripping down existing organisms like mycoplasma mycoids to the bare essential genes required for self-replication, sometimes utilizing only a few hundred genes, demonstrating the incredible robustness and redundancy built into even modern genetic systems. Conversely, chemists are synthesizing novel systems, such as the use of peptide nucleic acids, or PNA, which use a protein-like backbone instead of sugar phosphate to carry genetic information, testing whether life can emerge using fundamentally different chemistry. Early results show PNA can form stable duplexes and even interact with RNA, suggesting alternative chemical architectures for heredity might be viable in different planetary environments. The question of design, as hinted at by Einstein, might be interpreted not as deliberate intelligent planning but as the intrinsic property of physical laws to generate complexity when provided with the necessary thermodynamic gradients and raw materials. Life is the most complex chemical dissipative structure known to us, a system that actively lowers its own internal entropy by exporting heat and disorder into the environment, sustained by the constant flow of energy, the sun's radiation or geothermal heat. In this sense, life is simply the universe's most spectacular mechanism for dissipating energy gradients. The impossibility is overcome by the vastness of time and the relentless pressure of thermodynamics acting upon chemistry balanced precariously at the edge of stability. The sheer scale of the necessary assembly required for even the simplest living entity underscores the gulf between non-living matter and life. If the watch analogy holds, the components must not only assemble but must also incorporate a mechanism to repair, maintain, and duplicate the assembly process itself, using energy sourced from the environment, all while maintaining structural integrity against corrosive forces like water and radiation. The leap from the stable, albeit complex, self-assembly of fatty acid vesicles to a system capable of performing enzyme-mediated replication and energy harvesting is the true discontinuity in the history of our cosmos. It is the point where physics graduates into biology, point whose precise mechanism remains perhaps the most compelling, tantalizing mystery left to unlock in our exploration of the natural world, a mystery that forces us to confront the delicate, improbable balance that allowed the colossal, pre-wound clockwork of the universe to eventually produce an observer capable of contemplating its own improbable inception.